Cette conférence va maintenant être enregistrée.
So I'm glad to present you like the first webinar of this year for the RFMF. So the concept this year is slightly different from previous ones. The idea will be that young researchers that join the French community for their whole life will present us who they are, what they are working on, and what are their plans and vision about the future. So you can know more about them. And first speaker for this webinar will be Maud Heuillet, and she will present us her work as a research engineer in 13C Fluxomique. So Maud, the floor is yours.
Thank you. So hello, everyone. So I'm Maud, it's me in this picture. So I'm a research engineer in MS-based Fluxomics. And since September 2024, I'm a permanent research engineer at INRAE in Toulouse. So the most beautiful city in the world, as you can see here. And my main mission is to develop single-cell metabolomics in the MetaTool Fluxomet platform. So I'm going to explain how I ended up here. So to begin, a long time ago, I was a student at the University of Toulouse, and Jean-Charles Portet was my teacher in Métabolomique and Fluxomics. I think that's why I work in Métabolomics now. So for my master degree, it was a master in structural biology, metabolomics, and proteomics, focused on analytical chemistry and structural and functional biochemistry. For my internship, was in Switzerland, in Nestlé, and I developed a metabolomic study of liver lipids in diabetic and non-diabetic rats by a C13 approach. So I developed GCMS methods, but also some LCMS analysis, a little bit of NMR statistics. So it was really interesting. And at the end of my study, I acquired a knowledge in biochemistry, proteomics, and metabolomics, and also developed a strong interest in the study of metabolism. And I decided to continue in research by doing a PhD. But my master degree was a professional master degree and not for research. That's why I couldn't do a PhD in the University of Toulouse. That's why my PhD was a CIFRE PhD with private laboratory. So the LNE for Laboratoire National de Métrologie et d'Essai, under the supervision of Vincent Delatour. And the objective of my PhD was to develop reference methods for lipid profile biomarkers and apply these methods to quality control in clinical biology. So this PhD was in collaboration with an INSERM laboratory in Dijon. And the reference method is methods at the top of the pyramid here. So the most traceable method used then to calibrate all the other methods used for the industry and the testing laboratory. For example, the clinical biological laboratory. Voilà. So I developed GCMS reference methods. I calculate uncertainties. I purify lipids and lipoproteins. I organize an inter-laboratory trial with a lot of biological laboratories in France, so 40 laboratories. And I produce a reference material. So at the end of my thesis, the positive I took away from it, so I have become more independent and I have developed strong skills in project management because the thesis is a huge project. Metrology has also forced me to be more rigorous, but I don't want to pursue a career in metrology, but more in a functional approach of the metabolism. But as a researcher or as an engineer in the private or in the academic sector, I had no idea. So life is full of opportunities. So in 2014, I had the chance to join the MetaTool platform in Toulouse for a 2 years contract. So I was hired by Florian Beller, just here. And my job was to develop C13 Fluxomique analysis at MetaTool Fluxomique. So if we take a quick step back, Metabo Hub in 2014, so the coordinator was Dominique Rollin, and the objective was to develop a method in Métabolomique and Fluxomique, and also to establish the infrastructure in France. So Metabo Hub was 4 sites, one in Paris, Clermont-Ferrand, Bordeaux, and Toulouse, and it was around 80 people. And the MetaTool, the director was Jean-Charles, 4 different laboratories. One, it's MetaTool Metabolic Network, now it's Fluxomique. In MetaTool, we were 36 engineers and technicians with different specialties, so analytical chemistry, bioinformatics, mathematics, etc. And regarding the equipment, just here, we performed mostly triple quadruple analysis for the mass spec. We had 3 NMR spectrometer and 1 robot for the culture. On the Fluxomique platform, we used to provide, and we still provide, services for our clients. So, for example, macrokinetic data, so growth rate, consumption rates, or production rates of metabolites, mainly performed by NMR. We also quantify metabolites, so absolute quantification with NMR and mass spec, and also a C13 profiling. So it's carried out by tracking the incorporation of a C13 tracer in metabolites. And in some cases, go as far as calculation of metabolic fluxes. And I was involved in the metabolite quantification and C13 profiling by mass spec. So now to work on a platform, so the first aspect, so it's as part of a team, so it's a family spirit, as you can see here, it's the family spirit 12 years ago. So we work in close interaction with people from a variety of different fields, so NMR, MS, bioinformatics, robots, etc. So we all work in joint projects, so it's a day-to-day collaboration. And we are really committed to training and passing on knowledge. So I had some trainees, like Loïc Mervant, who is speaking just after me. In platform, quality assurance is also really, really important, even if it's not really fun. So the platform was certified ISO 9001 in 2014. And now we are audited twice a year. All the staff in the platform contribute to the quality assurance. I contribute all the day with the traceability of my project and my developments. And I contributed also for the health and safety and for the communication parts. As a platform, services and collaboration are core businesses. So I was responsible for managing numerous projects, which involved discussion with the clients, proposal, experimentation, reporting, and invoicing. Collaboration allowed me to develop specific methods for new class of compounds. And I was also in charge of specific methodological developments for the platform. So I decided to present 3 developments published. So here, the first one is the development of ISOCore. It's a software previously developed by Pierre, and we modify this software to take into account the data acquired on high resolution mass spec. So it's this software is really important for isotope tracing experiments, because with IsoCore you can correct your data for a natural abundance of isotope. Another development is a methodology for the validation of isotopic tracer analysis with mass spec. And the last development I did is a method to be able to quantify and to make isotope profile analysis in the same sample. And for that, we produce a specific internal standards produced by E. coli, where all the metabolites were labeled with C13 and nitrogen 15. So my first 2 years at Metatool were really interesting. And one thing was certain: I wanted to be an engineer in the platform, and if it was possible to stay at Metatool. And my contract were extended by 4 years. It was really nice, largely thanks to the collaboration we had with the Cancéropole Grand Sud-Ouest. So I was in charge of the coordination of the cancer and metabolism group, and I developed a lot of methods to measure isotopic profiling in mammalian cells. I was also in charge of the coordination of the internal projects in the platform, so organize meeting, lead this meeting. I was in charge of the external communication during external congress, and also in charge of the management of the project in health field. Things really interesting was the design and the implementation of a training course in C13 Fluxomics, where a lot of people of the platform were involved. It's a 1 week training, and we proposed this training since 2016, I think, from until now. So here it's the communication about our last training course this year. And I also have a lot of great collaboration with researchers in the cancer field, as Laetitia Linares and Laurent Lecam in Montpellier, with a lot of really nice publication. So after 6 years at Metatool, it was a really nice experience. I really wanted to continue at Metatool, but it was really complicated due to the research in France, so short-term contract. It was 6-year Metatool, so it's a maximum duration of contracts in the public research. And I didn't have any permanent position. So I applied to various positions in the academic sector, in the CNRS, and with INRAE. But I was placed on the reserve list, so it was really complicated for me. But opportunity again, I have been recruited in 2021 by Evotech as a research scientist in Métabolomique on a permanent contract. So briefly, presentation of Evotech. So it's a German biotechnology company founded in 1993 and specialized in drug discovery. It's a big company, and almost 800 people work in Evotech in Toulouse. So I was recruited as a project manager in Métabolomique in Antoine Berthemy's team. And this is the mission I had. So I participate to proposal with the client. So I write technical proposition and cost. I was in charge of the analytical development. So it was targeted approaches with a triple quadruple for metabolites and lipid analysis. And I write reports of results and also present my results with the clients. So the positive point, so it was a permanent position. It was really nice with a higher salary than in public, of course. But it was really difficult for me because I I don't feel really good at work because I needed more cutting edge developments and more scientific challenges. So it was a difficult time in my career because I was living in Toulouse with my family and my children. But I decided to resign to return to Metatool to work. So it was in 2023. So I start a 2 years contract for the implementation of single-cell metabolomics, hired again by Florian Valvert. And the good news was that I had a permanent position in 2024. So now just a few words on Metatool and MetaboWeb now. So now the coordinator is Fabien Jourdain, and 2 more sites join MetaboHub, Tours and Grand Ouest. So now the infrastructure is robust and include, for example, 100 publication and 300 projects per year. For Metatool, the chief now is Justine. We add 2 more sites, MetExplore and Fluxovivo, and we are more than 50 people with a lot of new instruments, really nice instruments. So now we have 12 LC HRMS. We have 4 robots, 1 for culture, but also 3 for the sample preparation and 1 cell sorter. We have MS imaging and a lot of software to help us to interpret our data. So in this slide is the Metatool scientific objectives we had. So it's a very challenging program. So we want to study the metabolism from the single cell to the whole organism. And now I will present briefly how am I in this objective, our work. So I'm involved in studying the metabolism of the whole organism, where this organism is represented by a fermenter. And in this context, I developed an online approach where we couple the fermenter directly to the mass spec. So the objective is to have a functional study of the metabolism. So as you know, metabolites are the final step of biological regulation, and metabolic changes can occur very rapidly. And also the fermentation monitoring is a pivotal tool for bioprocess optimization and control. So that's why we develop this kind of approach. So we couple a bioreactor to the mass spec, thanks to a lot of valve and pump and program. So here is the design in the lab. So it's a system of online metabolomics by MS, but the objective is to couple also a benchtop NMR to perform this kind of analysis. And we work on a suspension cell and adherent cell to have access to the metabolites in the supernatant and also inside the cells. And in parallel, we developed a real-time integration and reprocessing workflow. So why I decided to show you this project, because it illustrates what I like about working on a platform. So it's a technological challenge and also a teamwork. So Nina, just here, now lead the project, and she's in charge of the culture and the MS part. We are working really closely with Carl Burgess from the University of Edinburgh, and we took our inspiration from his work. Xavier here and Christophe are specialists in fermentation in the SOFERM lab at TBI. They help us for the technical setup and also for the automatization of the control of all the system. Rochelle, our bioinformatician, work on the workflow to have an automatization of the processing of the data in real time. Edern is in charge of the benchtop NMR part. And Nathalie helps us through the HepatoTwin project she leads on online metabolomics analysis of hepatic cells in which we are heavily involved. So it's a really interesting project. Regarding the cellular ecosystem, I'm in charge of the separation of the species grown in aquaculture. To do this, we purchased a new equipment. It's a cell sorter from Selenium, a Selen 1. It's a single cell sorting for the isolation and also a nanoliter dispensing. The advantage of this technology, so it's stressless. It's a piezoacoustic droplet generation, so it's ultra gentle cell isolation. It's really fast, accurate, and we can process any samples from 0.5 to 80 micrometers. So this sorter was installed in our lab last September, and it's a joint purchase with the platform Proteo tool in Toulouse because they use the Selen 1 for single-cell proteomics. So it's briefly an image-based technology, so we can isolate the cells in function of their morphology, so diameter and elongation, and we can also use a fluorescence marker. And because it's nanoliter dispensing, we can develop a lot of workflow, so the isolation, but also the sample preparation, add buffer, incubation, etc. So it's a device open for service. And it's really interesting to use this equipment because we have numerous collaboration and we can develop our skills for sortings. And we use this sorter to separate cell population from aquaculture. So it's a work performed with Cecilia Berges. And here it's an example of a project we had. So it's an ANR project led by Florian Belver. So it's micrometabo. Here it's Eden, who is in charge of the culture. And the objective is to separate two species, Acetobacter pomorum and Lactobacillus plantarum, and then analyze the isotopic profile of each strain separately after the co-culture, and to determine the optimum quantity to be able to quantify metabolites. But it's really complicated because, as you can see here, the elongation and the diameter are really similar. So it's really difficult to separate them based on the morphology. That's why now we try to do this using fluorescence, so different markers in function of the species. So this work is in progress. And now the last development I will present, so it's a big one. It's a development of a single cell approach from scratch. So my objective is to improve the sensitivity to better understand the metabolism, to have access to metabolic information in small populations, for example, in rare immune cells. We have a lot of projects with this kind of cells. It's really interesting also to study the heterogeneity of the metabolism and to measure to have access to the cellular metabolome of individual cells. For example, to understand the variability due to cell-to-cell interaction. And it could be useful also to perform high throughput screening approach, for example, to screen bank of mutants or screening drugs. But how I'm going to try to answer that, so it's a big question. So here it's the different step for really important for metabolomics, and it's the same for single cell metabolomics: the sample preparation, the MS analysis, and the data processing. So regarding the MS analysis, we want to have the most sensitive detector in the lab to be able to measure the metabolite inside just one cell. So we purchased a new equipment, the most sensitive mass spec of the market. So it's a 2-year work because we wanted to be sure of making the right decision, because it's a really huge investment. So Thermo lent me an Astral for 1 year, so I could carry out a lot of tests. And I was really lucky to work closely with their application services team in metabolomics and also in proteomics for all the nano-LC parts. And I learned a lot. I also organized a test of the Apemaldi source. So it's a source for imaging in collaboration with Gilles Frache and Mastec. So it was really interesting to plug for the first time this Apemaldi source on an astral, because we can use the high resolution of the orbit trap and also the speed of acquisition of the astral. So we had a lot of nice images, but I cannot show you the images because it was performed by my colleagues in the platform specialist of MS imaging. And in the same time, we talk a lot with the application laboratory in Bremen, in Germany, with Brücker, to make some tests with the timsTOF Ultra IP mass spec. We talk a lot about the development possible on our samples and also how we can make some modification on the source with the R&D service to optimize the copying with a microfluidic device. And it was a really difficult choice, but we choose the timsTOF Ultra IP mass spec, mainly because the source was sealed and was closed to prevent any contamination, because it's really critical for single cell analysis. And also because of in the timsTOF, we have the ion mobility, which is really interesting for us. So the installation of the timsTOF was last September. So the first approach I am developing is an offline approach. So I use the Selen1 to isolate the cell from the culture, and then I analyze the cells offline by mass spec. So I isolate a lot of cells, so big cells, so it's ELA cells, then yeast and E. coli, and at different quantity. Then with the Selen1, I can quench and extract the samples, and after the evaporation, the resuspension is performed by the nano-LC. And I developed two kinds of analysis: in direct injection, so flow injection analysis, and also with the nano-LC to have a good separation and to have a better detection. But it took almost 1 hour per sample. So now I am optimizing all the parameters to be able to analyze both lipids and polar molecules. And the last development, so it's an online approach, so to be able to couple a miniaturized culture to the MS detector. And between the culture and the MS, we will plug a microfluidic device to be able to send the cell one by one in the mass spec, and also eliminate the metabolites of the medium. And of course, to do this as fast as possible to avoid any metabolic changes. And to do that, I use all the work we performed for the online MS project I presented earlier. So to do that, we need to manipulate the cell one by one. So it's really complicated and it's not my specialty. So to do that, I make a lot of collaboration with experts. So for example, Pierre-Joseph at the LAS, so he's a biophysics and microfluidic expert. So he helped me to design microfluidic chips to do what I want to do. Also Michael Maucourt in Metabo Hub Bordeaux, also an expert in design, production and microfluidic chip. And to finish, Michael Castelin, it's a researcher at TBI in Toulouse in our lab. He's a physicist and an optical tweezers expert. So he built for us a system using optical tweezers to manipulate the cells. So here it's the principle, so it's not my specialty, so it's it will be really fast, but we use a highly precise laser to grab and move the cells by creating a radiation force that attracts the object on the most intense point of the light. So here you can see the scheme. So the energy is transmitted to the particle during the reflection, and the laser just here attracts the cell to the center of the beam. and push it in the direction of the beam. So it moves like that. That's why we focus the beam to immobilize the particle just at the center of the z-axis. So with the laser, we can catch the cell and immobilize the cell. And in the real life, it looks like that. So the laser beam is deflected by mirrors until it reaches the sample just here and here, where it passes through the objective lens, which focuses it onto the sample. There is also a LED above to illuminate the microscopy area, a camera just here, and a fluorescence detector. All of this allows us to perform a conventional microscopy without the laser being switched on. So we can perform classical microscopy observation and also use the laser to catch the cell with the optical tweezer. For now, everything is controlled by an Xbox joystick. Hop, on va m'entendre parler, je crois. So here you can see in red is the laser, so we can move the cells with the laser and make a pétanque with the other cells. So it's really, really impressive and it's really nice. And now the automatization is in progress to don't have to use Xbox joystick, but to make it completely automatized. And to finish, this is my work now. So I manufacture a microfluidic chip like this. Here is a wafer with silicon. Here is the chip I use to isolate the cell. It's this kind of chip. Here it's a plasma. It's used to put the PDMS on the cover glass. Here is the results of my experiment yesterday. So as you can see, it's a beautiful channel clocked, so it doesn't work. And here it's in the lab, so the optical tweezer. You can see the cells and the mass spec just here. And here it's to all the pump for the microfluidic devices. So it's really, really nice work, but a lot of work to do. So to conclude this presentation, so what's a research engineer in the platform? So you work a lot with the team. It's a lot of service development for scientific community, both academic and private. It's cutting edge instruments and development. It's really, really interesting and challenging. In a platform, we promote research, so we write a lot of scientific articles and participate to international conferences. For example, last year for the Métabolomique and the RFMF. So it's a lot of challenges, but also a lot of fun. So thank you very much. If you have any question.
So if you have a question, you can also open your microphone and ask, or you can also write in the chat and Maud will answer. So does anyone has a question now? Otherwise, I do have many.
Thank you.
So first, I have a general question about your vision of organization. So you work in a research platform, and I know that it's relatively independent in terms of structure from any other research team. So how do you organize the research part of your work and also like the service part in your platform? Like, what is your vision about that?
For me, it's I do services, so I do analysis for researcher, of course, but I'm very lucky because the R&D part is really huge and a lot of work to do, so I don't do a lot of services. But in the platform, everybody do both services for the researcher and R&D, because it's really important to have money to can work and make some research and development.
If I can intervene, do you think when it's going to be finished, the development of the single cell, you're going to have more services to do, as it will be a routine analysis, hopefully in a few years?
Yeah, I think we will have a lot of collaboration with single cell, and I hope we can make a lot of it. But now tomorrow, because it's a really, really huge work. But yeah, I think the objective is to open the single cell analysis to the community and make a lot of services about that. And the objective in long term is to perform fluxomic analysis in one cell, but not tomorrow.
I had a follow-up question on Loïc ones, because like you're really working on like cutting-edge instrument and you're developing new instrumentation, and I think that's extremely nice. And you kind of have the power to shape what kind of analysis will be done in France in the future by developing those new methods and those new instrumentation. So I had a question on how those directions are decided and how do you envision who will use it within the community? Will it be mainly local? Do you envision some international collaboration and so on? So how do you plan when you say, okay, we will go to do some single cell metabolomics, and then you go into that project and you take several choices?
Oh, it's really complicated to understand because I don't know now, but we plan to have a lot of collaboration. Collaboration first with our lab TBI, because we have a lot of projects on heterogeneity. So it's really important for these people to have access to a single cell analysis. Also in health domain, all the collaboration we had for Cancéropole Grand Sud-Ouest, a lot of researchers are interested in a single cell analysis in health. For the international collaboration, I don't know yet, but if I made some presentation in Congress, we could have other collaboration. I think it's really interesting. A very few laboratories develop single cell approach now. So I think it will have an impact on the collaboration in the future.
And I have another one. Since you pile up a lot of tools to manage to get to your endpoints, is that thought that those tools, like the optical tweezers, the single cell sorter, and so on, and also like the team stuff, would they be also open to a marginal amount of time for doing some other niche analysis that they could unlock? Or the idea is that all those tools together, they synergize to a single cell and they will fully be dedicated to single cell in the future?
Regarding the cell sorter, it's used for single cell metabolomic, but also for a lot of different projects because it's open to the community. So other researchers in the lab use just to make sorter, to separate different kind of cells, not just for metabolomics, for transcriptomic, for a lot of things, for proteomic also. The optical tweezer will be dedicated to single cell because it's really, really specialized analysis, of course, but not just for this kind. We plan to use it with a lot of projects, with a lot of collaboration. Regarding the because the optical tweezers can be used, but with this device we can also make classical microfluidic coupling. So this will be also just for single cell, but also we plan to use this to work on organoid also, to have more signal. And the team stuff for now is dedicated to single cell because we don't want to put a lot of cells inside to have an impact on the sensitivity of the mass spec, because we want a really, really clean machine.
And about that, I had more technical questions because you said that, I mean, I know it's only the beginning of the single cell, but you have a project where you try to separate two different bacteria in co-culture. And you think that in the future you will be able to separate more complex culture, like involving like maybe 5-10 different cell types? Or do you think it's not now?
To separate the cells with the CLN1, it's possible to separate a lot of cells if they are different.
Mm-hmm.
You need to have different morphology, for example, because it's really complicated if the shape is the same or with fluorescence. But if we have different fluorescence, if we want to isolate one cells from a lot of different species, it's possible, for example, if it's fluorescence.
So you could imagine doing like a surface marker for different cells.
Yeah.
Sort them like a bit of flow cytometry before and go into the mass spec.
With the with the CNRS.
Yeah.
Not the fax. Yeah, it's possible, and then isolate in the plate and then couple it to the mass spec. It's possible.
And also, I think someone has a question.
Yes.
Yeah, keep going. I will ask later. Don't worry.
Yeah, and you show lipids detection in single cell analysis. And what about metabolites? Do you see them, or is it easier with lipids?
For now, in my first test, I just saw lipids because it's easy.
Yeah.
Sure. Now I try to optimize everything to be able to measure metabolites. But in FIA, because it's an injection from the non-LC, I inject 1 microliters, the samples are really diluted. So it's really complicated to see metabolites. And the objective is to analyze directly in the cells with the microfluidic to don't have any dilution, to inject the cell in the system, to list the cell, and to analyze directly everything enter in the mass spec. So we hope it will be easier to detect something without any dilution.
Yeah, so I have a question regarding to your FIA method. So did you do a lot of like development for this method, or like, you know, which criteria did you evaluate, etc. when you use the FIA method for your single cell metabolomics? Yeah.
Which can you just repeat how
like,
I made the method?
Yes. Did you like optimize the method, or you know which criteria did you use to evaluate your method, etc.? And did you compare your FIA with the nano LC columns?
With the FIA method, I just optimize. We perform this with Q-Exactive, with an Explorer, excuse me, for the online approach, the first development. And the detection, we perform a lot of scan range to optimize the signal. I didn't perform this on the cutoff. And how I validated the methods, I just inject different quantity of cells, and when the cell rise something in the cells. And I optimize on the, and regarding the MS optimization, I optimized parameters on the mix of standards, both metabolites and lipids, to have the highest. It's really complicated because everybody enter in the same time in the mass spec, so it's not really sensitive. Mm-hmm. And is to compare this with really fast. In 1 minute, we have the response, so it's 1 minute. It's really fast, but the objective is to validate it with the nano-LC. We have the separation to confirm the identification we. But it's ongoing.
Okay, thank you.
So let's all thanks Maud again for her presentation. That was really nice. And now the next presentation will be given by Loïc Mervant, that has the right to share his screen now and has been recently also hired as, if I'm not wrong, a researcher at INRAE. Loïc can correct me if I'm wrong, and he will present us also his background, what he will do, and his vision about it. So Loïc, the floor is yours.
Thanks. Do you see the screen correctly?
Yes.
Okay, good. No, you are right. I just been hired at INRAE as a researcher. So today I'm going to try to present my past experience and how they made who I am now, I guess. So I would like to thank you for the invitation, but also I honestly hesitated to say no because I thought it was difficult to be interesting talking about my life and my scientific life. But yeah, as you said, I'm a newly recruited researcher, so I started in February this year, so it's very, very new. I do mass spec metabolomics since a few years, and if I have to define myself, I would say that I'm interested in good stories and how to make sense about of metabolomics data. And today I'm going to try to exemplify how over the course of my scientific path, I try to add a bit more biology at every step to get more complete stories. So I started metabolomics the same way as Maud did, actually, doing the master called BSPM in Toulouse University, and a master that provides knowledge in structural biochemistry, proteomics, and metabolomics. And I think for the same reason, because I had Jean-Charles Porté as a teacher, I was really into mass spec metabolomics, and I wanted to do a first internship in mass spec metabolomics. And so I was lucky enough to find an internship at the TBI, Toulouse Biotech Institute, and I was supervised by Maud and Florian. So it feels weird to share the stage with Maud because I know I will forever be her pew-pew. But anyway, the topic of the internship was to try to develop an untargeted isotopic profiling method using LC-HRMS. So the internship objectives for me were to learn the metabolomics workflow from the sample preparation to data analysis. And we wanted to test various extraction methods, analytical method to maximize metabolite detection, and use workflow for metabolomics for untargeted processing of labeled data, and apply that as a proof of concept on biological sample. So this is just to illustrate what I've just said. So some people were doing the labeling experiment for us, and we were doing the metabolomics workflow with the hope of deciphering labeling incorporation in an untargeted manner. So not everything worked, I remember, in this internship. But at the end of it, I was pretty convinced that I wanted to continue in mass spec metabolomics. But I was also, I realized that I was also a complete ignorant on the biological part, and I wanted to learn more. And I was also convinced that I will never do a PhD, and I wanted to work in industry. So that's what I did first. I was hired as a research engineer in Saclay in Paris. I mean, in Saclay and Paris at Medday Pharmaceutical, supervised by Sandrine Arros, who is now the metabolomics platform leader at the Pasteur Institute. And the lab of the company were hosted in the CEA, in the drug and metabolism lab led by François Fenaille. And so that's where I spent a few years as a research engineer in metabolomics in the pharma company. In this job, I had three main missions. The first one was to do some cohort analysis on various samples. So we were doing plasma or cerebrospinal fluid with the hope of discovering biomarkers for neurological diseases such as multiple sclerosis or amyotrophic lateral sclerosis. Another part of the job was to perform some analytical development, so multiplex quantification for metabolites in spinal cord, for example. We also tried some things about isomer discrimination based on MS2 profile. And the last part was more targeted analysis to understand the mode of action of the drug, the Medday drug, which was a high-dose biotin on multiple sclerosis. So it was a really interesting job, but after a couple of years, I decided to quit for several reasons. So there were a few things I liked about this job, notably the cohort analysis part and the biological interpretation of the data. I really like to spend time looking at the metabolite list. I liked also the fact that a significant proportion of the job was analytical development, and I think that really helped me to upgrade my skills in mass spec. And I have a bit of academic freedom, I would say, so I could join the RFMF Junior and participate to the first RFMF school in 2017. But there were a few things that I liked less and that made me quit the job. Notably the partial involvement in the project as a research engineer. So it's a bit the same as Maud said in her presentation. So as a research engineer, I was involved only on the analytical part, but had no vision on the entire project, especially at the beginning of the project, the design and the scientific strategy, where people with PhD had. So I was like, okay, maybe I should do a PhD to do that. And the other frustrating point for me was that we were doing only metabolomics. So we were looking at the metabolite, the significant metabolite generating maybe pathway enrichment, but we had no way to go further into the hypothesis. And this was, I thought it was a bit frustrating. So in the end, I was like, okay, maybe why not to do PhDs that try to combine biology and metabolomics. So back to Toulouse, I started a PhD at INRAE Toxalim, so where I am now, in the team I am actually now. So between two teams, the teams led by Françoise Guéraud and Laurent Desbrouwer, so in between the metabolomics platform and a research team. And the PhD project was how to use metabolomics to better understand the link between red and processed meat and colorectal cancer. So the main reason why I chose this PhD was because there was a good proportion of analytical development, and I wanted to continue to have my hands on the mass spec and to upgrade my skills. The main part was because it was aiming to do metabolomics and combine it with biology. So for example, with in vivo work or in vitro model to get a more comprehensive picture of what happened. And it was a good opportunity as well to upgrade my skills and knowledge in biology. And the last part was because we got our hand on a cohort analysis. So it was really interesting for that. And when I started my PhD, my mindset was, okay, let's do a PhD and go back to industry. And I will never do a postdoc. That's not for me. So in this PhD, I had three main axes. The first one was analytical development. We wanted to optimize and develop a method allowing for the exploration of urinary metabolome and allowing to do a focus on electrophilic compound, because the whole theory behind this PhD project was that red meat is generating oxidative stress. And we can monitor oxidative stress with some specific metabolites in urine. So that's what we wanted to achieve here. The second part was the human and in vivo cohort analysis. So here we really wanted to study the impact of red meat consumption on the metabolome and try to combine the result we could have in the cohort and in the human cohort and the in vivo cohort and other kind of analysis. And finally, we wanted to complete those data with biochemical and physiological data to have a more big picture, I would say. So I will talk a bit about this cohort analysis. So we basically started by analyzing the cohort. So it was the Nutrinet Santé study, and we had 240 samples of people eating more or less red meat. And so when we performed targeted metabolomics on these samples, we actually didn't have a lot of metabolites significant. So I remember it was maybe 10 or something like this. But a good proportion of them were derived from the microbiota, which led us to think that maybe red meat consumption is impacting microbiota, and we could actually correlate or just look at the data we had on an in vivo cohort that we performed in the lab, where we fed rats with a high red meat diet or high white meat diet. And we realized that we had actually the same trend and the same significant difference on the same metabolites, which helped us to strengthen the hypothesis of something happening on the microbiota. And because on rats we can do more analysis, we could go for microbiota analysis. And we could link the metabolomics data and the microbiota because we realized that we observed an increase in the species producing indoxyl sulfate and a decrease in the ones that were producing cinnamoglycine. And just to talk about these two metabolites, indoxyl sulfate is a uremic toxin and generally is associated with the bad health of the microbiome, while the cinnamoglycine is in general associated with the good health of microbiome and associated with a good diversity and everything. So the hypothesis was that red meat was some kind of deleterious for the microbiota. And we were happy to have concordant results between human and in vivo. And the last thing we noticed is that the microbiota signature was suggesting that it was a signature of microbiota that had experienced oxidative stress. So we looked at some genes in the colon to look at this oxidative stress response, and we looked at some genes that are some defense genes for oxidative stress, and we observed that we had an increase in expression of these genes in the colon tissue of the rats. So if we put everything together, what we were able to propose was that red meat and heme iron, which is why red meat is pro-oxidative, generate ROS, and that will impact the microbiota. And that's why we can have, we can see significant metabolites, microbiota-derived metabolites in urine. And in parallel, these reactive oxygen species are increasing the expression of antioxidant genes. And it's not enough to replenish the levels of the main antioxidant body, which is the glutathione. Here we did MS imaging on the colon of the rats, and we saw that the glutathione signal in the colon were really lower in the red meat group compared to the white meat group. And so this is how, by linking different omics and different data, we were able to propose a bit more complex story. So what I like in this PhD was actually this mix between the analytical chemistry and biology and linking different data sets and omics to strengthen the observation. I also like the freedom to build a bit my own project and to be involved in scientific networks. So that's where I joined the RFMF Junior, for example, and also a first experience in managing. So I had 2 master students, for example. But there were a few things I liked less in this PhD. So because we were working on electrophilic compounds, we were very limited in our annotation capabilities. Also, the roller coaster effect of the PhD, where you can combine very exciting and frustrating times, and the thesis writing was really painful, I found. After this PhD, I was not convinced anymore that I wanted to work in industry, and maybe continue on that way to try to combine metabolomics and biological model. So I went for a postdoc, this time in London, in Karen Vousden lab at the Crick Institute. So it was a P53 and cancer metabolism lab. So the main reason why I chose this postdoc was because the advertised project was on obesity and pancreatic cancer using metabolomics. But in the end, it turned out that my postdoc project was do what you want related with pancreatic cancer, because this project was already given to other postdocs. So it was kind of a surprise, interesting. But it was actually a new challenge, and with more biology, still using MS metabolomics. It was a cancer metabolism lab using isotopic labeling method. So it was I was happy because that's what I did in my internship. It was an international environment with a lot of different people and a prestigious institute with many facilities. So just about the institute I was in, it's called the Francis Crick Institute. It's nice building just here. We were more than 2,000 people in it. There was more than 20 scientific platforms ranging from metabolomics, genomics, advanced imaging platform, or everything you can think of. And there were especially dedicated to researchers. There was no external collaboration with these STPs. And there were about 100 research labs on various topics. And I think that place was actually, I was very impressed at the beginning working in this environment. But I think that helped me to grow as a researcher. So after spending a bit of time of getting lost in the building and getting lost in trying to find a postdoc project, I finally ended up with two projects. The first one was to understand the crosstalk between the pancreatic tumor and the metastatic site, something called the pre-metastatic niche. And the second project, actually, I have to thank my friend Mark Hennekar, who was a former postdoc in the lab and a senior postdoc when I arrived, and he helped me to get involved in the team, and I joined him on his project of the role of this enzyme on the early step of the pancreatic carcinogenesis. Today I will just talk a bit quickly about the multiomics project. So basically the core concept of this project was to study the pre-metastatic niche. So the hypothesis is that having a primary tumor, the primary tumor is doing something to the metastatic site, here the lung, even before the circulating cells arrive to the metastatic site. So the idea is that primary tumor is maybe secreting some factors or doing something that prime the tissue to be more amenable to the circulating cells and to facilitate the establishment of metastasis. So I had to learn new models to do that, and I had to learn how to do ultrasound on mice. So we were basically doing injection of pancreatic cancer cell line in the pancreas of mice. And after a few weeks, and to let the tumor grow, we were collecting a whole bunch of samples, so ranging from plasma to lung tissue, and we were performing metabolomics and proteomics on these samples. So we ended up with a lot of data set that we tried to integrate, and we did several mice cohorts to strengthen the observation as well. And when we did the integration, it revealed that the top hits were actually from the same compartment in the metabolism. It was from the arginine metabolism. So we observed a massive decrease in the arginine levels and an increase in this enzyme, arginase 1, which catalyzes this reaction, arginine, to forming urea and ornithine. So that was the first observation we were able to make. And that was the center of our story. So multiomics revealed something about arginine metabolism in the pre-metastatic lung. And so from there, we could ask some questions. So for example, was this observation specific to the lung? So we went back to the data, and the answer was actually yes. So we looked at the substrate and the product of this reaction. And in the lung and in different cohorts, we were able to see the same thing, like a decrease in arginine and an increase in the product. It was true for the lung tissue and the lung interstitial fluid, but it wasn't true for the plasma, so suggesting that this effect was kind of local. We also looked at other metastatic sites for pancreatic cancer, especially the liver, and we didn't see any changes at all in arginine levels, suggesting again this local effect. The other question we wanted to answer was where does this arginase 1 increase come from? So how can we explain this burst in arginase 1? So for that, we had to look at the arginase 1 main producers, which are a cell type called the myeloid cells, which includes macrophages, dendritic cells, neutrophils, and monocytes. So to do that, we performed some flow cytometry on the lung tissue to analyze the cell population in the pre-metastatic lung. And we realized that we have a massive increase of the neutrophil population and the monocytes population. We confirmed this observation using immunohistochemistry. And so that was our link to arginase 1. So we have more arginase 1 because we have more neutrophils and monocytes in the lung. But then the next question was, how can it be mediated? So why are these cells coming to the lung? So for that, we looked at a bunch of inflammatory cytokines, and we realized that we had a lot of inflammatory cytokines in all the compartments. And so these inflammatory cytokines are known to be chemoattractant for neutrophils and monocytes and myeloid cells in general. So that could explain why we have so many in the lung. But the last question was, what can be the consequence of all of that? And this arginine 1 increase, arginine deprivation in the lung. So we know that arginine is actually an essential nutrient for T cells. And the T cells are the immune cells that fight tumor cells. So the model for now is that we have inflammatory cytokine that can recruit myeloid cells and induce proteome and metabolome alteration and inducing arginine deprivation. And when we try to do some proliferation assays on T cells in vitro, we realized that a low arginine condition was actually preventing T cells to proliferate. So we confirmed that arginine is really essential for T cells, and that a drop in arginine could impair the function of T cells. And so that's the whole story. We were able to propose that the pre-metastatic lung is a low arginine environment that could impair T cell efficiency, and that's why maybe it's easier for the cells to establish a metastatic niche in the lung compared to other organs. So what I liked in this postdoc was to discover a new environment and a lot of opportunities to learn. I also liked the challenge of creating a project from scratch on a new topic, and team up with lab members with different expertise, because honestly at the beginning I was a bit lost, and it was really interesting and helpful to have people to rely on and to integrate the team. There were a few things I liked less though, especially the publication pressure, because it still seemed like the only way to move forward in academia, and everyone was super focused on how to publish as fast and as high as possible. I also liked less the difficulty of creating a project from scratch on a new topic, because it was both a challenge and a difficulty. And it was as well a relatively competitive environment, even though it was interesting. So towards the end of the postdoc, I wanted to settle and have more time to create long-term research project. So that's why I ended up wanted to continue in academia and as a researcher. So I was lucky enough to be recruited at INRAE Toxalim. The advertised position was a researcher on dietary transition and cancer risk, with a focus on plant-based and diet and pancreatic cancer. So basically the context of this subject is that in Western countries, we are going to do a transition toward more plant-based diet. And this is going to redefine the dietary exposure and potentially the impact on human health. So as we increase the plant-based diet part in our diet, we are more exposed to some contaminants that are harmful to humans. So for example, cadmium, pesticides, and mycotoxin. But we are also exposed to beneficial compounds such as polyphenol, antioxidant, and fibers. And this is this risk and benefits that we want to assess on pancreatic cancer, because there are few evidence in the literature that especially cadmium can be linked to pancreatic cancer. And it's a very trendy topic. As you maybe seen a few weeks ago, we had a lot of press release on cadmium and diet in the French media. So the general objectives of this project will be to assess the benefit risk of a plant-based diet on the pancreatic cancer. We aim to define biomarkers of exposition and effect of the different contaminants we're going to select. and we want to generate knowledge on the mechanism of pancreatic cancer promotion impacted by the different contaminants. And we also have a more long-term objective to participate to the establishment of a nutritional recommendation, for example, or to be able to propose biomarker to extrapolate to humans and help to with the biomonitoring, for example, monitor cadmium exposition in human populations. And the general strategy to do that is to study the exposure to identified contaminant using biological models of increasing complexity. So we will start with very simple models such as pancreatic cell lines, all the way up to pancreatic cancer models in vivo. And I think I will rely a lot on LCMS metabolomics. So the first part here will allow us to look for a specific cell-specific mechanism in response to the contaminants, while here we're going to look more to the impact of the contaminants on the progression of the disease, and also try to find some biomarkers of exposition to cadmium or biomarkers of effect, with the hope of applying all of this to a human cohort. So this is the plan. This is the general strategy, but now what's the plan? So I just started, so I don't have any data to show. And my first objectives are really, really short term. So I have to generate preliminary data in order to apply for grant. I have to trying to initiate collaboration with former and new colleagues to explore new areas. For example, setting up collaborative project to look at the impact of this contaminant on the immune compartment. Trying to get access to human cohort samples as well. We actually set up a PhD subject that you can have a look here if you are interested in doing a PhD maybe. But the good thing about being in academia is you have some kind of freedom, and I can work also on some side project. So as I told you, I'm really interested in how to process metabolomics data. So we are currently developing a new tool to look at metabolomic signature from a different perspective with Clément Freinet. And I aim to develop metabolomics on organoids as I did a lot of in vivo work in the past years, and I think it will become increasingly constrained by admin things, and we will be less and less able to use mice, and that's good because I don't really like it. So yeah, that's a plan to try to generate more complex cellular models and try to analyze them. So I think I will go and see MOD in the near future. And yeah, that's my plans for the future. And I would like to thank all the people I met during my scientific path. And this is my mentor board. And I would like to thank all the entire teams I worked with as well.
So thanks a lot, Loïc, for this wonderful presentation. So as I said before, if you have any questions, you can ask in the chat, and you can also raise your voice and ask the question directly. So while you recover and prepare your question, maybe I have some to start. So my first one would actually be, you spent some time abroad, and what would you like to bring back in your current research environment from your postdoc environment? And
the money?
Outside of the money, like more lab culture or stuff that you were not expecting to see, and you think that, yeah, maybe this is the way to do, or I would like.
Yeah, no, I was kidding. I mean, honestly, it was really different from what I experienced in France, and it was interesting to see both sides. I'd say that I liked the way that the lab meetings were, for example, where at every lab meeting, one postdoc has to present the project. So it was kind of a bit stressful sometimes, but I mean, we were seeing the advancement of every lab members, and it was interesting to have a nice overview of what everyone is doing. So for that, it was nice. Yeah, let me think about it. I don't know. Yeah, there was a few things. I mean, I really liked how the institute was structured as well. I mean, the platforms were dedicated to the researchers, so that was a really luxury environment to work in.
Hello, should I go on? Do you hear me well?
Yes.
Okay. Hi, Loïc. Thank you for the nice presentation. It was great to see your curriculum. I wondered if you could tell us more about the competition that you took for the researcher position, how it happened, how many stages, interviews, and what you were asked specifically, and if you have some good tips and advices to prepare for these kinds of selection.
Yes. So basically, this competition will depend where you go. So I know that at INRAE, you apply for a project that is already made. So the project was already defined, and they wanted to work on the plant-based diet and the risk of pancreatic cancer. So at INRAE, you have to show how you are a good fit for the project. And I know that, for example, if you go for INSA competition, it's going to be very different. You have to bring your own project and find a team that want to sponsor you to go to the competition and present your research project. So I can give advices about the INRAE one, but for CNRS or INSA ones, I would be a bit clueless. So there were two stages actually. The first one is you write your scientific profile and you have to explain according to your past experience how you are a good fit. You have to highlight your scientific achievements and everything. It's relatively short to write. It's like maybe 5 or 10 pages. I mean, it took me months because I really wanted to have a nice file, but it was in the end it's a short document. And once you reach that step, you are or not selected for an oral exam. So it's like a 45 minutes oral exam. You don't have, it's very strict. So after 45 minutes, the rings bell and you get out. You have 12-minute presentation to present how again you're a good fit for the job. And then there are 33 minutes of questions where you have to answer, I think, the most questions possible. It's really fast, but actually I found it quite interesting. But it's a bit impressive. You have like 15 members of the jury in front of you in a small room. So yeah, it was a very intense experience.
Thank you for sharing your experience. I have another question, but I will wait for my next turn.
No, you can go ahead.
Okay, then quickly the second one was about the cohorts, because yeah, you mentioned you want to get your hand, like your next objective is to get your hand on some cohorts, and I think of course that's important, and a lot of us are looking for these kinds of opportunities. So I wondered where and how you were looking and triggering these opportunities to take part into cohorts.
I have a few different strategies. So I was lucky because we were last week in contact with some people that are actually building a cohort on pancreatic cancer and environmental exposure, and they were the people who contacted us. So it was in this way it's very convenient. But I also plan to apply to the EPIC cohort, which is an European cohort on nutrition. And they have lots of samples, and it's a retrospective cohort. So people are enrolled in the cohort without any particular disease, and they monitor them over the years. And then you can select the one that developed pancreatic cancer, for example, in a year. So I think they have 300 or 400 patients. people that had pancreatic cancer. So my plan is to contact the people that are managing this cohort and beg them to send me some samples. But yeah, it's not an easy part. And the last thing I know is there are also a cohort you can just pay for to have access to them. So there is something called Backup, I think, in Toulouse. It's a cohort about pancreatic cancer again, and there you can. It's really expensive, but I think you can just pay to have some samples.
Okay, thank you.
I think, Fabien, you can ask your question now.
Oh, yes, thanks. So thanks, Loïc, for the wonderful presentation, and as well as Maud. So I have a bit the same question as Theo, but for the private sector. So what would you bring from the private sector to your strategy, both in the management and the research?
So I think it's a bit more difficult to answer to that one because I think it was a long time ago and I was more junior than now. But well, let me think about it. The thing is, honestly, I mostly retain the fact that the involvement of the project was really separated, and I don't think that's a good thing for academic research. So that I would not keep it. I don't know, maybe the way the project were managed, not in terms of people implication, but I think they were really defined, and there were a lot of go no go steps, and maybe sometimes it could be a good thing not to go into a project that will never work, that we can sometimes do in academia. So yeah, maybe set up some go no go steps more in academia would be sometimes helpful.
Okay, and I have another question. So you plan to use in your current project, you have Métabolomique quite involved in the project, and but it's Métabolomique the state of the art, or do you see some potential bottlenecks or new things that you will have to develop, and in which direction?
Yeah, the thing is, when I was preparing the presentation, I was trying to picture myself in a metabolomics landscape, and I realized that I will probably do a lot less analytical development as a researcher, and I think I will rely on people like Maud, for example, to develop super nice fancy tools to analyze organoids or single cells. But yeah, I mean, my goal is to still do a bit of metabolomics. So, for example, this organoid analysis, I would like to carry it myself, for example. Yeah, I think that there are a few adjustments to be made, but mostly I would say a good 90% of state-of-art metabolomics, and maybe depending on the project and where we go, do some analytical development. So yeah, I would be also very interested in the immediate response to the contaminant. So you add the contaminant and you try to track the cellular response in the few minutes and few hours. So I know typically the thing you develop with Natalie, the hepatotwin, where you can monitor in real time samples, that would be very interesting for this. But I think it's going to be mostly collaborative work than me doing the work myself. But maybe I'm wrong. Apparently I'm very bad at predicting the future.
You are never wrong, Loïc. Okay, thanks.
If no one has other question, I may have one last before closing this webinar. It will be about going abroad and then going back to the French community, because this is what is expected out of every postdoc, I have the feeling. And I think this is not that simple because my view is that the French community is still very self-centered. So what is your perception about that?
I think an important part is to keep your network and your connection while you're abroad. And this will be helpful at every step. So now I'm really happy that I stay connected with people from the Métabolomique, so it's easier to go back to them and say, hey, I'm a researcher now. Can we connect to maybe try to set up some project together? I think that's a very important step. And yeah, to do that, go to conferences, present your projects. I think that that's really helpful. But yeah, I mean, it's the postdoc step. If you want to go for a researcher position, I think it's now mandatory. So yeah, you have to do it. And honestly, I don't regret it. It was very nice.
So if anyone has an additional question to Loïc, feel free to ask now. Otherwise, I would thank you all for being here. And thanks again to Maud and Loïc for their presentation. It was really cool. And on that, I would wish everyone a good end of the afternoon and see you next time. Bye-bye.
Thanks.
Bye.
Bye-bye.